Welcome to biochemistry lecture. In today we are going to see the three important questions which have been asked in the previous university exam under carbohydrate chapter. First question is fat of pyruvic acid. Second, we will discuss how much amount of ATP molecule is produced during glycolysis process, and we will be detailing seeing about gluconeogenesis. The first question is fat of pyruvic acid. So we all know the end product of glycolysis is pyruvate or pyruvic acid. So the pyruvate is obtained from various substances like glycolysis through glycolysis process. Alanine, from alanine it is converted to pyruvate. Malic acid is also converted into pyruvate. Lactate is also converted to pyruvate. Similarly, cysteine and serine is also converted to pyruvate in various chemical reactions in our body. Next, we will see the what are the fate of pyruvate. So, when question is asked, you need to write the start from sources of pyruvate, then you write the fate of pyruvate. So, pyruvate can be converted into oxaloacetate, or pyruvate can again converted into alanine by reversible process. Pyruvate can be converted into acetyl CoA. You know that acetyl CoA enters the citric acid cycle. The lactate which is obtained from the pyruvate can reversibly converted into pyruvate again, and the ethanol can also be converted into the pyruvate can be converted into ethanol and malic acid. So this is the fate of pyruvic acid. Now, basically, in our textbook, they have given the how pyruvate is converted into acetyl CoA and how pyruvate is converted into lactate. So we are focusing only on the two things. First is the conversion of pyruvate to acetyl CoA. So under aerobic condition, under the presence of oxygen, pyruvic acid is transported into mitochondria by a proton that is called proton symporter. Okay, so proton symporter carries the pyruvic acid into mitochondria. So in the mitochondrial matrix, in the cytoplasm of the mitochondrial matrix, pyruvate undergoes oxidative decarboxylation. So what is happening inside the mitochondrial matrix? Pyruvate undergoes oxidative decarboxylation. By this process, the pyruvate is getting converted into acetyl CoA with the presence of an enzyme pyruvate dehydrogenase. With the presence of an enzyme pyruvate dehydrogenase. So hope you understood how pyruvate is converted into acetyl CoA. The next one is how this reaction takes place, that is represented in this figure. You can see that chemical structure is not much important in nursing point of view. If you are good in drawing the chemical structure, you can write. So during the conversion, what is happening? One AD molecule is reduced to NADH. So for that, what is happening? The carbon dioxide decarboxylation, carbon dioxide is released. So that is the process. So this reversible reaction is the link between glycolysis and citric acid cycle. So when what is the link between citric acid and glycolysis? The conversion of pyruvate into acetyl CoA is the linkage. Okay, now come to the next thing. How pyruvate is converted into lactate by reduction process. By reduction process, the pyruvate is converted into lactate with the help of the enzyme lactate dehydrogenase. The reduction of pyruvate by NADH to form lactate. That process is called reduction process. Pyruvate undergoes reduction by releasing the NADH. It is converted into lactate with the help of enzyme lactate dehydrogenase. This reaction takes place in the cells when the amount of oxygen is limiting us in muscle during intense activity. So usually lactate is not produced in our body when you do vigorous exercise, when you are having more activity level. During the time, the pyruvate is converted into lactate to give energy. For this conversion, the enzyme lactate dehydrogenase is very helpful. So that diagram is given here. So this is pyruvate. Pyruvate is undergoing a reduction process, and what is happening that NADH plus H plus is converted into NAD plus nicotinamide nicotinamide dinucleotide, and this is you are having lactic acid diagram in your right hand side, and pyruvate diagram is present in the left hand side. Okay, so most of the conversion takes place in our skeletal muscles. So because the skeletal muscles produce large amount of lactic acid in the hypoxic condition when you are doing any physical activity, excess amount of exercise. So body demands more amount of oxygen because less amount of oxygen is present inside your body due to the activities. So during that time, skeletal muscles produce lactic acid. So accumulation of lactic acid in your body produces severe pain. So you understood now what to write in the question for fat of pyruvic acid. Now let's see the next question. So how many number of ATP molecules are produced by glycolysis process? So basically the production of ATP in the glycolysis reaction takes place in two conditions. First condition is aerobic state and second condition is anaerobic state. So what is happening? What is happening in the aerobic state? In aerobic state, so in the presence of oxygen, so what is happening? NADH produced during the conversion of 3-phosphoglycerate to 1,3 bisphosphoglycerate enters electron transport chain, that is ETC, and thereby it releases 3 ATP molecule. Thus, additionally 6 ATP molecules are produced per glucose molecule. So this is how. So in the question, what examiner who is evaluating your paper wants from your answer sheet is this diagram. You must able to write. That is how much amount of ATP is consumed. 2 ATP molecule is consumed. How much amount of ATP is produced at substrate level? 2 into 2, 4 ATP molecule is produced. At the oxidative level, so we got 2 into 3, that is 6 ATP molecule is produced. So in aerobic state, the total yield under aerobic condition is 8 ATP molecule. The net yield is 8. That you can see it clear here. The ATP molecule consumed, that is in the energy production investment phase. In the energy investment phase of glycolysis, what is happening? 2 ATP molecule we are utilizing to the reaction to occur, and in the energy obtaining phase, harvesting phase, the energy harvesting phase. So at substrate level, 2 molecules of ATP is produced from each substrate. Okay, so that is totally 4 ATP molecules produced. Similarly, 3, what is happening? 3 ATP molecule is produced from the two the reaction agents. So totally 6 plus 4 10. Already we have 2 consumed. So total is the net gain for us 10 minus 2, that is 8 ATP molecule we are getting. So this is in the aerobic glycolysis. We are getting 8 ATP molecules. So what is happening in the anaerobic state? In anaerobic state, NADH, the NADH produced cannot be used by electron transport chain for ATP production. Why? Because there is lack of oxygen. So in the absence of oxygen, the NADH which is produced cannot be used for ATP production by the ETC, that is electron transport chain. So less ATP production is happening in the anaerobic stage when compared to aerobic glycolysis. So in anaerobic state, we are having less number of ATP is produced. So let's see how it is produced. So this tabular column is very important in your answer sheet. So ATP consumed is, we know that 2 ATP molecule is consumed, and ATP molecule produced at substrate level 2 into 2 4. At oxidative level, what is happening? We are getting 6 ATP molecule. So the total is, we are having totally, leave the NADH. So after leaving NADH, we are having only ATP, 4 ATP molecule. We are having only 4 ATP molecule. So this oxidative level is not taking place because it is in anaerobic condition. So we have only 4 ATP molecules. So the net gain is 4 minus 2, that is 2 ATP molecules. So number of ATP molecule which we are getting in the aerobic stages 8. In the anaerobic stage, just we are getting 2 number of ATP molecule we are getting. So this is the answer for ATP production in glycolysis reaction. The next important question which we are going to discuss in this video is gluconeogenesis. Gluconeogenesis means the synthesis of glucose from non-carbohydrate sources. The site where it takes place mostly in liver. Around 1 kilogram of glucose per day is produced in the gluconeogenesis in the liver. Also, this occurs in kidney, and particularly the site where exactly it is happening is cytosol of the cell. And what are the substances needed for gluconeogenesis? The precursors. First one is pyruvate, second one glycerol, third is lactate, fourth propionate, fifth glucogenic amino acids. In this video, we are going to see only how the glucose is produced from pyruvate molecule. In the next videos, we will see how the glucose is produced from other substances like glycerol, lactate, propionate, and amino acids. Now let's see the steps in pyruvate conversion to glucose. Before saying that, we want to know why we need glucose. So glucose is needed for various reasons. The first thing, the body has to maintain the normal range of blood glucose level. So for that we need glucose. If glucose level goes to hypoglycemia, there is low blood glucose level, then liver will start producing the glucose from the non-carbohydrate substances. And essential organs and vital organs like brain, kidney, erythrocytes, RBC needs continuous supply of glucose in the form of energy for its function. Skeletal muscles, whenever it gets tired, it needs glucose for its rapid action. The skeletal muscles, particularly at the time of exercise, have less oxygen. As a result of that, the amount of glucose is utilized faster. So we get tiredness. So to overcome that, glucose need to be supplied to the skeletal muscles for its good function. At the time of fasting, glucose level will become low. So the gluconeogenesis process produce the glucose and it maintains the basal requirements of our body. Now let's see the steps in gluconeogenesis. Gluconeogenesis takes place in 3 steps. Basically, the gluconeogenesis is, the steps in glucogenesis resembles the glycolysis pathway in a reverse order. So the reverse order of glycolysis will be gluconeogenesis, but some steps are different. There are 3 points where the reactions are differs. The first one is the conversion of glucose to glucose 6 phosphate with the help of the enzyme hexokinase. The second point, where fructose 6 phosphate is converted to fructose 1,6 bisphosphate. The third one is phosphophenol pyruvate convert into pyruvate with the help of pyruvate kinase. So these are the 3 reactions or steps where it is have some difference comparing to the glycolysis. The first step, the conversion of pyruvate to phosphophenol pyruvate. So the glycolysis, the last step we are getting pyruvate. So that takes place here. In the first step, what is happening? The pyruvate is converted into phosphophenol pyruvate, but this does not take place in a single step. It takes place in two steps. First, the pyruvate is converted into oxaloacetate with the help of the enzyme pyruvate carboxylase. Pyruvate carboxylase helps to convert pyruvate into oxaloacetate. The oxaloacetate thus formed will be converted into phosphophenol pyruvate with the help of the enzyme phosphophenol pyruvate carboxylase. The oxaloacetate will be converted into phosphophenol pyruvate with the enzyme phosphophenol pyruvate carboxylase. So this is happening in the step 1. The step 2, so you can see the first step, the oxaloacetate is converted into phosphophenol pyruvate, and the second step, up to fructose 1,6 bisphosphate, the steps are same like glycolysis. So the phosphophenol pyruvate which is produced in the step 1 will undergo various changes, and it comes to the product fructose 1,6 bisphosphate. The in between the steps are same like glycolysis in the reverse order. Okay, now the phosphophenol pyruvate will undergo change, and it comes to fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate will be converted into fructose. This fructose 1,6 bisphosphate will be converted into fructose 6 phosphate. Fructose 1,6 bisphosphate will be converted into fructose 6 phosphate. For this, the enzyme catalyzing is fructose 1,6 bisphosphatase. Fructose 1,6 bisphosphatase, but that takes place with the help of magnesium ion. That takes place in the support of magnesium ion. This conversion takes place. Fructose 1,6 phosphate is converted into fructose 6 phosphate. The next step, the last step, the glucose 6 phosphate, which is produced in the second step, will be converted into glucose. Glucose 6 phosphate will be converted into glucose. So these are irreversible reactions which takes place in the glycolysis. Now what is happening? This glucose 6 phosphatase, the enzyme will catalyze the glucose 6 phosphate for its conversion into glucose. So this takes place in the liver and kidney, but this reaction does not take place in the muscle. So in the muscle, there is different mechanism which will occur for production of glucose, that we will see later. So keep in mind this step occurs only in the liver and kidney. Liver helps to supply the blood sugar through gluconeogenesis. So in the last step, glucose 6 phosphate is converted into glucose with the help of enzyme glucose 6 phosphatase. So this is what gluconeogenesis, the conversion of pyruvate into glucose. Hope you have enjoyed this video. If you have any doubt, you can ask your queries in the comment box. I will try to answer it. In the next video, we will see how other substances are converted into glucose. Take care. Bye bye.